Welcome students to an in-depth exploration of biomembranes, where we will unravel the structural intricacies, dynamic properties and vital functions of cellular membranes. Today's discussion will connect foundational concepts like the fluid mosaic model and membrane asymmetry to advanced research and clinical applications, equipping you with the knowledge to understand and investigate membrane biology at a postgraduate level. Our learning objectives are: analyze biomembrane composition and component functions, to evaluate properties driving membrane dynamics, to interpret the fluid mosaic model and its evolution, to investigate mechanisms and implications of membrane asymmetry, apply functional implications, to relate advanced concepts to clinical research and therapeutics. Let us start with the introduction to biomembranes. Biomembranes are indispensable structural and functional units of life. Biomembranes, the dynamic scaffolds of life, are vital for all cells, serving as selectively permeable barriers, signaling hubs and regulators of biochemical processes. Every cell, whether prokaryotic or eukaryotic, is bounded by a plasma membrane, while eukaryotic cells further contain internal membranes that define organelles such as the nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus and lysosomes. These membranes act as selectively permeable barriers, regulators of biochemical processes and platforms for communication, without which life would not exist. Biomembranes are composed primarily of lipids 40 to 50%, proteins 50 to 60% and carbohydrates 1 to 10%. Their properties like fluidity, permeability and asymmetry enable cells to maintain homeostasis while responding to environmental cues. First, take a look at historical perspectives of cell membrane. Biomembrane studies evolved from early microscopic observations and models to molecular level analysis. A landmark in this journey was the Denali-Davidson model in 1935 that proposed a static lipid protein sandwich, but the fluid mosaic model by Singer and Nicholson in 1972 revolutionized this view, depicting membranes as dynamic with fluid lipids and mosaic proteins, which shifted the view from static bilayer to dynamic mosaics of protein and lipids. Let us take a look on composition of biomembranes. The composition of biomembranes is a masterful blend of lipids, proteins and carbohydrates, each contributing to the membrane's structural integrity, functionality and adaptability. Although the basic design is conserved, variations in the relative proportions of components confer specific properties and functions to different membranes. First are lipids that form the bilayer foundation and constitute the bulk of membrane material. They are amphipathic molecules possessing hydrophobic, that is non-polar hydrocarbon tails and hydrophilic, that is polar head groups, enabling them to self-assemble into bilayer in aqueous environments. Major classes of membrane lipids are phospholipids, the most abundant class derived from glycerol or sphingosine. Glycerophospholipids, for example, phosphatidylcholine, phosphatidyl ethanolamine, phosphatidylserine, phosphatidyl inositol. Phosphatidylcholine, often called lecithin, is prevalent in eukaryotic membranes, promoting bilayer stability due to its cylindrical shape. Their polar head groups interact with the aqueous environment, while Fatty acyl chains contribute to the hydrophobic interior. Sphingolipids derived from sphingosine backbone. Example include sphingomyelin, glycosphingolipids and gangliosides. Sphingolipids such as sphingomyelin add complexity featuring a sphingosine backbone and contributing to lipid rafts, that is microdomains enriched in cholesterol and sphingolipids that serve as signaling platforms. Next is cholesterol, a sterol with a rigid ring structure and small polar hydroxyl group. Cholesterol intercalates between phospholipid tails, modulating membrane fluidity. In animal cells, cholesterol can comprise up to 50% of lipid content, preventing phase transitions from fluid to gel states at low temperature and reducing excessive fluidity at high temperatures. Next are glycolipids. Glycolipids with carbohydrate moieties are primarily on the outer leaflet, aiding cell recognition and adhesion. Next are the proteins. Membrane proteins are the workhorses of the membrane, accounting for diverse functional capacities. Types of membrane proteins. First are integral membrane protein or intrinsic membrane protein. They span the lipid bilayer, that is why they are called as transmembranes, or are embedded within the layer. It facilitates transport, signaling and enzymatic activities. Examples include ion channels, receptors and transporters. Second type of proteins are peripheral or extrinsic proteins. They are loosely attached to membrane surfaces via non-covalent interactions such as spectrin in erythrocytes attached via electrostatic interactions. They provide structural support and mediate interactions with the cytoskeleton. Examples include cytoskeleton proteins, signaling adapters. Third group of proteins are lipid anchored proteins. These proteins are covalently attached to lipid molecules, providing membrane association. Example include GPI anchored proteins. Now let us look at functions of proteins. Proteins help in transport of molecules and ions. They help in signal transduction. These proteins help in enzymatic activities and they also help in cell-cell adhesion and recognition. Now let us move on to carbohydrates. Carbohydrates accounts for 2 to 10% of the plasma membrane proteins. They are covalently linked to proteins called as glycoproteins or covalently linked to lipids called glycolipids and are usually oriented towards the extracellular space. The carbohydrate functions include cell recognition, immune response and receptor ligand interactions. Glycocalyx, a carbohydrate rich outer layer that shields cell from mechanical stress and pathogens. Now let us move on to properties of membrane components. First, physical and chemical properties of lipids. Fluid and phase. Lipids enable lateral diffusion modulated by unsaturated fatty acids, while cholesterol buffers fluidity stabilizing cells. Phase separation forms lipid rafts for signaling, with conical lipids like phosphatidyl inositol aiding fusion and cylindrical lipids like phosphatidylcholine stabilizing bilayers. Protein properties, mobility and functionality. Proteins exhibit lateral diffusion with amphipathic integral proteins, example rhodopsin, facilitating transport and signaling. Post-translational modifications like palmitoylation target rafts, while crowding 30% membrane area limits mobility in bacterial membranes. Next, carbohydrate properties, specificity and protection. Hydrophilic carbohydrates like glycans on the outer leaflet provide specificity via diverse structures mediating recognition. For example, selectins in leukocyte rolling and pathogen repulsion via sialic acid's negative charge. Amphipathic nature, self-assembly. Amphipathic lipids and proteins self-assemble into bilayers with hydrophilic heads facing water and hydrophobic tails forming the core. Hydrophobic interactions, bilayer stability. Hydrophobic interactions between lipid tails minimize water contact, stabilizing the bilayer interior. Membrane dynamics, diffusion and flip-flop. Lateral diffusion is rapid, flip-flop is rare but facilitated by flipase scramblase for asymmetry maintenance. Let us move on to the fluid mosaic model. The fluid mosaic model, proposed in 1972 by Singer and Nicholson, remains the cornerstone framework for understanding biomembrane organization, portraying membranes as dynamic fluid systems with embedded proteins. It portrays membranes not as rigid structures but as fluid, heterogeneous and functional platforms where lipids and proteins interact to regulate cellular processes. The fluid mosaic model remains the most accepted framework for describing membrane organization. First, let us talk about historical context and development, origin and evolution. The fluid mosaic model integrated biophysical and biochemical data to explain the lateral mobility of membrane components. Key features of the fluid mosaic model are bilayer and fluidity. The lipid bilayer forms a fluid matrix enabling flexibility for vesicle budding and protein movement. Flip-flop events maintains asymmetry, example phosphatidyl serine inner side, phosphatidyl choline outer side, and ensures functional specialization of leaflets critical for signaling and polarity. Mosaic arrangement. Integral proteins, example ion channels like sodium potassium ATPases, and peripheral proteins, for example spectrin, are asymmetrically embedded, creating a mosaic pattern. Their dynamic interactions with the cytoskeleton and extracellular matrix form complexes that regulate processes like cell adhesion and signal transduction. Role of cholesterol and lipid rafts, regulation and platforms. Cholesterol intercalates between phospholipid tails, buffering membrane fluidity and preventing excessive rigidity or disorder. Lipid rafts concentrate signaling molecules, example G protein coupled receptors or GPCRs, serving as platforms for efficient signal transduction in immune and neural cells. Flexibility and dynamics. Membranes can bend, invaginate and form vesicles. Membranes exhibit remarkable flexibility capable of invagination driven by proteins like clathrin, vesicle formation and shape changes during migration, division or endocytosis. This flexibility supports processes like phagocytosis in immune cells and neurotransmitter release in neurons. Membrane fluidity is essential for protein mobility, lipid diffusion and dynamic interactions between macromolecules supporting transport and receptor ligand interactions. It underpins dynamic processes like membrane repair and cell migration critical for wound healing. Now let us move on to membrane asymmetry. One of the striking features of biomembranes is asymmetry in composition and function. First, let us talk about asymmetry in lipids. In outer leaflet, it's enriched in phosphatidylcholine and sphingomyelin, whereas on the inner leaflet, it's enriched in phosphatidylethanol-amine, phosphatidylserine and phosphatidylinositol. In case of protein, the orientation of receptors, channels and enzymes is highly specific. N-terminus may face extracellular space while C-terminus faces cytoplasm. In terms of carbohydrate asymmetry, they are exclusively extracellular, contributing to glycocalyx, essential for immune recognition and pathogen host interactions. Functional consequences of asymmetry are cell polarity and vesicular transport, regulation of signaling cascades, programming cell death and recognition. Now let us move on to functional implications of biomembrane organization. Biomembranes are not just physical barriers, their precise organization supports diverse cellular activities essential for survival. The asymmetric distribution of molecules enables membranes to act as dynamic platforms for transport, signaling, communication and remodeling. Transport mechanisms. Biomembranes orchestrate molecular movement critical for cellular homeostasis through diverse transport mechanisms. For example, passive transport. It includes simple diffusion, for example, oxygen and carbon dioxide across lipid bilayer, and facilitated diffusion via channel proteins, for example, aquaporins for water. It relies on concentration gradients without energy input. Number second, active transport. It uses ATP driven pumps like sodium potassium ATPase to establish ion gradients, essential for nerve impulses and nutrient uptake. Endocytosis and exocytosis involves vesicular trafficking. Endocytosis engulfs extracellular material, example, phagocytosis of pathogens, while exocytosis releases molecules like insulin. These mechanisms ensure selective permeability, maintaining cellular environments. Signal transduction. Biomembranes serve as signaling platforms, detecting and relaying extracellular cues to regulate cellular responses. Membrane receptors such as G protein coupled receptors, example adrenaline receptors, and receptor tyrosine kinases, example EGFR, bind ligands to initiate cascades like CAMP or MAP kinase signaling, controlling growth and metabolism. Lipid rafts concentrate signaling molecules, enhancing efficiency. These pathways are critical in development and disease like cancer, where EGFR mutations drive uncontrolled proliferation. Next is cell-cell communication. Membranes facilitate interactions between cells, enabling tissue formation and signaling. Adhesion molecules, for example, cadherins, and junctional complexes, for example, tight junctions, maintain structural integrity and signaling as in epithelial barriers. Synaptic transmission in neurons involves neurotransmitter release across synaptic membranes mediated by voltage gated channels, crucial for neural communication and brain function, with disruptions linked to disorders like epilepsy. Membrane remodeling. Membranes dynamically reorganize to support cellular processes like migration and division. Endocytosis, vesicle budding and membrane fusion driven by proteins like clathrin and snares enable nutrient uptake and secretion. Dynamic reorganization during cell migration, for example, lamellopodia formation and cytokinesis, example cleavage furrow, relies on lipid asymmetry and cytoskeleton interactions, critical for tissue repair and embryonic development. Now let's discuss advanced concepts on biomembranes. Biomembranes as dynamic cellular interfaces are at the forefront of cutting-edge research, revealing novel insights into their nanoscale organization and therapeutic potential. Clinical implications. Membrane defect-driven diseases are cystic fibrosis. Cystic fibrosis transmembrane conductance regulator, in short CFTR. It is a colloid ion channel protein located in epithelial cell membranes. Mutations in CFTR impair chloride and water transport, causing cystic fibrosis, which leads to thick mucus buildup in lungs, pancreas and other organs. Next, spherocytosis results from defects in cytoskeleton proteins such as spectrin or ankyrin, leading to loss of membrane stability and fragile sphere-shaped red blood cells prone to hemolysis, causing anemia and splenomegaly. Lipid metabolism disorders such as Niemann-Pick disease involve defective sphingomyelin degradation, leading to lipid accumulation, disrupted membrane integrity and impaired cellular function. Membrane proteins like human epidermal growth factor receptor 2, in short HER2, in breast cancer. It is a receptor tyrosine kinase belonging to the EGFR family. Overexpression or amplification of HER2 drives aggressive cell growth and proliferation, particularly in HER2 positive breast cancer, making it an important therapeutic target. Drugs like trastuzumab. In conclusion, dear students, we conclude our exploration of biomembranes. You have gained a profound understanding of their composition, the transformative fluid mosaic model, and the critical role of membrane asymmetry in cellular function. Multifunctional platforms central to cellular life, signaling and homeostasis. These insights equip you to tackle advanced research questions, from unraveling disease mechanisms to developing innovative therapies in biotechnology and medicine. Carry this knowledge forward to make meaningful contributions to the dynamic field of membrane biology. Thank you. See you in the next lecture.